Thank you everyone for joining today. Welcome back to our tech seminar series. For those of you who don't know me yet, I'm Alex Lemens, the director of our Molecular and Genomics Informatics core. We also have today, Pat, the director of our Genomics Center, and the team from Nanopore, who'll be talking about their awesome, I'm a big fan, they're fun little sequencers. And I do also want to point out that the Genomics Center does now have both the GridION and MinION, which Stephanie and the team will tell you about more later. But just so if anybody wants to do these types of projects, we do have the availability to do them already. But before I turn it over to the team, just the general housekeeping items. If you have any questions, please drop them into the chat or the Q&A box. We'll monitor it for anything that we could either answer on the fly or anything that's not answered during the seminar, we will then bring up at the end of the talk to ask at the end. But with that, I'll turn it over to the Nanopore team to hear more about the awesome, fun little Nanopore sequencers.
Thanks, Alex. And good afternoon, everybody. And again, thanks to the Magic and Genomics Center for inviting us to speak today. We'll be providing an introduction to nanopore sequencing, the applications that are possible with the technology, and the recent advancements that we've made. So my name is Stephanie Acosta, and I'm the Senior Inside Sales Representative for the Northeast. And then I'll also pass it off to my colleagues, Monalina and Aaron, to give an introduction to themselves.
Do you want me to start? So hi, everyone. I'm Monalina. I am the account executive for Northeast. Before joining, I just recently joined the sales team, but before joining, I was a application scientist for almost 7 years at Nanopore. So yeah, I have kind of a bit of technical knowledge, obviously, but learning the sales process right now.
Hi, everyone. I'm Aaron Pomerantz. I'm a market segment manager and happy to be here. I've been with Oxford Nanopore for a little over a year now, but I started out my journey with Nanopore technology back when it was first released in 2014 and have been using the technology ever since. So happy to be here, and I'll describe some of the specific applications for some microbiology. So thanks.
Yeah, so at Oxford Nanopore, our goal is to enable the analysis of anything by anyone, anywhere. And at the heart of our technology is, of course, the nanopore. So here's a cross-section of the pore and how it's embedded into the flow cell membrane. An electrical current is applied within the flow cell, and the motor enzyme, shown here in purple, feeds single strands of DNA or RNA through the pore, causing disruptions in that current. And these disruptions are what we call squiggle data, and they're acquired in real time and stored as FAST5 files. Then our base algorithms come in and translate these squiggles into a sequence of bases, and these base called files will be delivered in a FASTQ format. So this motor protein is actually part of our sequencing adapters, which are attached during the library prep. So the main function of this protein is actually to slow down the translocation speed of nucleic acid through the pore so that it can be properly read. Without it, the molecules would pass through at a rate of around 1 million base pairs per second, which, as you can imagine, is extremely fast, and it would be pretty difficult for our algorithms to make sense of that signal. So we've slowed down this translocation speed to around 400 to 450 base pairs per second. And there's many advantages to sequencing this way. With Nanopore, you have the option of preparing sequencing libraries via a PCR or PCR-free manner, allowing a native look at either DNA or even RNA instead of a copy. And with PCR-free options, base modification information is preserved and can be directly interpreted without the need for bisulfite conversion. And when it comes to read lengths, the sky is the limit. Technically, there is no maximum fragment length because we put you in control of this. So whatever your fragment lengths are, that will equal your read lengths. And with short fragment mode now integrated in our Minnow software, we're able to sequence fragments as small as 20 base pairs, which is really great for projects where you're looking at cell-free DNA, circulating tumor DNA, and even FFPE samples. And the current customer read length record is 4 megabases. And the rapid real-time data collection allows you to only collect the data that you need. So you can stop your sequencing run at any time point, flush the flow cells, and reuse them for another sample. The real-time nature of the tech also enables a new generation of targeted sequencing to be possible. So enrichment or depletion of your target can occur at the software level without any upfront targeted library prep needed. This feature is called adaptive sampling, and we'll definitely dive into this a little bit later. As a result of these benefits, our data is very information rich. So from a single experiment, meaning a single library prep, you can perform SNP and structural variant detection, look at methylation patterns, phase, and even assemble genomes. On the accuracy end, we've made great improvements over the past few years. We currently offer the highest single raw read accuracy on the market at Q23 with our latest chemistry updates. And most recently, we were able to achieve a perfect 100% accurate single read at 72 kilobases. Now, this is a great slide because the data shows the progression of where we've been and where we are now. So at the bottom, we have our R9 simplex data, which has been our gold standard chemistry for a while. And at the top, we have our new Kit 14 duplex data. From this, you can see that our duplex reads have very few errors. And in duplex mode, which is when the complement and the template, when the complement strand is immediately sequenced after the template strand, the single read accuracy for this is over Q30. And we have a growing variety of kits to best fit the scope of your experiments. So whatever your application, we have a kit to suit your sequencing needs. We're the only technology to offer prep-free software-based targeted sequencing and a native look at DNA and even RNA strands without the need for reverse transcription. PCR is always optional, which allows you to obtain methylation data without any chemical treatments. So if we just go from left to right on this table, our ligation sequencing kit is our standard double-stranded DNA kit that can be used for genomic DNA, amplicons, cDNA, and the prep only takes about an hour. And we do have multiplexing capabilities for up to 96 samples on a single flow cell. And our rapid sequencing kit is transposase based, meaning that it uses a transposase complex to simultaneously cleave and attach our sequencing adapters to your fragments. With this kit, you can also multiplex up to 96 samples at a time, and it's actually the basis for our plasmid sequencing workflow, which we'll discuss later as well. And then we have our ultra-long kit, which is actually a modified version of our rapid kit that produces ultra-long reads with N50s of over 50 kb. But if you're unable to meet the input requirements for the PCR-free preps, we do have PCR options for samples with very low input. And for RNA experiments, the direct RNA kit sequences the native RNA strand without the need for PCR or reverse transcription, which means that you can look at RNA modifications as well with our technology. However, if you're not interested in base modifications for RNA, there are a couple of other options that offer higher outputs, such as the PCR cDNA kit and direct cDNA kit. Both require reverse transcription, but the direct kit is PCR free. And then on the targeted side, we have a full-length 16S sequencing kit that targets the V1 through V9 hypervariable regions, and a kit that's also very unique to Nanopore, which is Cas9 enrichment. And this kit offers a PCR-free way to enrich for your regions of interest. And then, as we've mentioned many times already, we have a software-based enrichment technique called adaptive sampling, which is sample prep free. And then next, we'll move on to some of the applications. So we'll start with methylation, whole genome sequencing, and targeted sequencing with Oxford Nanopore. So as some of you may have heard, earlier this year, Ewan Ashley and his team at Stanford University set a Guinness World Record for the fastest DNA sequencing technique. This story started when a 13-year-old boy was airlifted to San Francisco Hospital with a critical heart condition. And along with the physicians, they were able to extract, prep the sample, sequence, and analyze the data within 7 hours and 18 minutes. The analysis pipeline they used was able to produce Q-rated VCFs with annotations within 5 hours and 2 minutes. So that time frame is actually included in the total 7 hours and 18 minutes because the team was able to begin analyzing the data just within 15 minutes of sequencing. So the sequencing and analysis happened simultaneously. Traditionally, rapid characterization of disease-causing variants from whole genome sequencing has been challenging. And whole genome sequencing enables better detection of such variants, but has typically taken days or weeks to return a result. So this is an incredible feat. And now I'll pass it off to my colleague, Monalina, to kind of give us a little bit of a background on our methylation sequencing and how it compares to bisulfite.
Awesome. Thank you, Steph. So I'll start with this, and I love saying this. So regards to methylation detection, we are not better than bisulfite sequencing. You probably already heard, if you're using Nanopore already, we integrated Remora. It's a tool to enable real-time epigenetic insights with Nanopore sequencing software, and Remora is outperforming on all precision recall, accuracy, and F1 scores. So in terms of bisulfite, it cannot easily differentiate between methylation types such as 5-MC, 5-hydroxymethyl-C, or add the detection of other modification types such as 6-methyl-A, but Remora that we have integrated can. So yeah, nanopore sequencing, it opens up the possibility of looking at methylation directly on individual DNA molecules and RNA molecules, and therefore, with regards to DNA, it represents how the DNA strands were in the cell, potentially the best, we say is the most true way of looking at DNA. So if you look at, and Stephanie, since she's sharing the screen, will guide through an arrow. So here's some methylation benchmarking data. As we explained before, with nanopore sequencing, you can sequence native DNA and native RNA and read modified epigenetic bases without the bisulfite conversion and PCR. So that means that you can access like really challenging regions of the genome, for instance, like repetitive or low complexity regions. Also, if you look, it gives you more uniform mapping across the genome, which is shown here on the left side of the graph. And also on the right side, if I draw everyone's attention, it shows that no bisulfite and also no PCR. It means that with ONT, you get higher mapping rate, and additionally, the analysis time is significantly faster. Next slide, please, Steph. Thank you. So just kind of talking about why are studying base modifications important. So one example would be epigenetic characterization of cell-free DNA. And this is an emerging approach for characterizing diseases. And also, first, why do scientists study cell-free DNA? So circulating cell-free DNA, it can reveal information, informative features of like tissue of origin. It can you can look at somatic genome alterations, DNA modification, and cell type specific fragmentation patterns as well. So jumping off of that, DNA methylation is a promising cfDNA biomarker, and if you look at some latest publications, also widespread testing as a cancer screening tool. So aside from cancer, methylation pattern can also be used to detect the turnover of damaged cells in a time-sensitive condition, such as in terms of a myocardial infarction, sepsis, or even COVID-19. So specifically on this slide, this paper on cancer methylation profile from cell-free DNA, it came out of Stanford University from Billy Lau's team. So Billy Lau and his team had a few challenges they wanted to address that I want to list. So one is characterizing cancer-specific methylation changes. It's proven to be very highly sensitive and specific approach for liquid biopsies and cancer disease monitoring. But characterizing cfDNA methylomes from patients remain very challenging, particularly with when you look at conventional sequencing approaches like bisulfite GC bias. You can have low yield or just short reads and not being able to characterize it correctly. And also cfDNA is often lower than like about 1 nanogram per milliliter. That's and also for base modification studies, PCR just isn't an option, right? So because it will otherwise remove those modifications. So the team's aim was to design a nanopore-based workflow to enable the characterization of cfDNA methylome as a means of liquid biopsy for cancer monitoring. So they used our PromethION 24 device to generate the data. So with nanopore sequencing, they were able to look at native DNA methylation calling. It enabled the analysis of methylome over their cfDNA without obviously PCR amplification. And also, because they didn't have to do any PCR, they looked at native DNA methylation. There was no PCR-associated GC bias. With their modified protocol, they were able to generate sequencing libraries from like nanogram amounts of or less of cfDNA per sample. So addressing kind of the issues that you might have with low cfDNA yields. And with flexibility in library prep, they were able to do multiplexing with their samples, so that also reduced their cost. So overall, this approach would stand out by being PCR and bisulfite free, and it would also generated hundreds of millions of reads per cfDNA sample, like an order of magnitude improvement over just previous studies that they have done. And of course, it also supports its use for longitudinal monitoring because it requires nanogram amounts of cfDNA per sample. So if we look at the next slide, So thanks, Steph. Continuing on, so they observed a longitudinal trend that correlated with specific clinical events based on the analysis. This is probably like a sad example, but extremely important for cancer detection and characterization. So for one patient who was receiving treatment for metastatic colorectal cancer, they sampled blood over 600 days. So if I could draw everyone's attention to the figure on the right, the upper panel is showing total cell-free DNA reads, and lower panel is showing methylation profiles matching those of the matched tumor. So after having undergone chemotherapy and surgery, the patient here had a period of stable disease, but then starting from like day 400, the fractional reads with tumor-specific methylation changes dramatically increased. So this change also correlated with the CT imaging they did, which showed substantial metastatic progression in multiple organs. And gene-level methylation analysis of this patient's longitudinal cfDNA also showed gene-level methylation changes found in the matched tumor. So overall, this approach has a potential to impact liquid biopsy diagnostics for cancer detection and characterization, including relapse and metastasis. So let's go. Thank you. So there are two more examples here of how base modification studies are important. Since just recent, past understanding of how even methylation manifests were very different with what we understand now. So scientists at Hebrew University of Jerusalem, with collaborators from Canada and Italy, also studied cell-free circulating tumor DNA from patients with lung cancer and some other cancer. So a lot of, in the paper, they describe a lot of perception in the community was as cancer progresses to different stages, there is a hypomethylation. But that is clearly not the case with what we saw in a previous example. They did see, in many cases, depending on the gene, the methylation patterns that there were some hypomethylation. For this group of scientists, a Nanopore platform provided a portable and rapid genome sequencing segue, and its ability to natively profile DNA methylation with complex sample processing was, for them, an attractive point of care real-time sequencing. So they demonstrated that ONT shallow whole genome sequencing, you were able to detect copy number alterations from circulating tumor DNA of cancer patients. They also showed that cell type and cancer-specific methylation changes were also being able to detected, as well as cancer-associated fragmentation signatures, as you can see on the diagram. So this feasibility study suggests that ONT shallow whole genome sequencing could be a powerful tool for liquid biopsy. And so let's look at another example on the left side. This is from Winston Timp's lab at Johns Hopkins University. So when you apply this to a different location, it might be interesting that motor regions of different genes shown here in cancer, you see different patterns of methylation. So what's plotted here is normal thyroid genes, early metastasis in thyroid cancer, and then if you go down, late metastasis in thyroid cancer. So what you see here are individual sequencing reads where methylated DNA is red and unmethylated DNA is blue. So the concentration of blue you see in the promoter of human telomerase reverse transcription gene, that's where the concentration is of the methylation. So there's a big CpG island that is usually unmethylated in normal thyroid. And what they see is an erosion of that unmethylated region of the promoter region. So as you go more into the late stages of metastasis. So another interesting thing that they observed is in the late metastasis, you have a mutation in the allele itself. So if you look at what we labeled as MUT and then what we labeled as the wild type. And then the methylation pattern in the wild type, the mutant allele has more unmethylated islands, because this is really interesting because you see phase mutations along with epigenome on those reads.
And then now for my favorite part, adaptive sampling. So adaptive sampling allows for targeted sequencing to happen at the software level, which alleviates the need for you to enrich for your target upstream of library prep. Before sequencing, you would upload a bed or FASTA file of your reference sequence. And because of the real-time base calling, when the DNA strand enters the pore, our Minnow software will start aligning the sequence to the reference that you've uploaded. So it will check the first 400 base pairs of any molecule that begins to translocate through the pore to see if it matches the reference file. And if it is a match, it'll continue sequencing that strand and move on to the next one, which you can see in the image on the bottom right. But if it doesn't match, the strand will be ejected from that pore, as shown right above that image on the top right, and that pore will also be freed up for another strand. So you can either choose to enrich for your reference sequence or deplete it, which becomes very useful for projects where you may have a lot of host contamination and you're looking for a way to deplete the host genome without having to incorporate that step into your extraction process. And as standard, you can also identify any base modifications using this method. And a couple of adaptive sampling studies are shown here, such as the one by Dr. Kavaka and others at Johns Hopkins. In this project, they sequenced a 148 gene cancer panel to a depth of just 30x on a MinION device and identified twice as many structural variants as short-read sequencing, and with 100% concordance to deep hi-fi or even deep nanopore coverage of reads. Another example is the adaptive sequencing of MHC, and we were able to achieve 20x coverage across the entire 5 megabase pair region within just a few hours. Another interesting study on this slide is the BRCA target enrichment. So we started the run sequencing BRCA1 and depleting BRCA2 for the first 20 hours. And then we switched and depleted BRCA1 and enriched for BRCA2. So this example just shows the power that we have to manipulate the sequencing runs using adaptive sampling. And I'll pass it off to Monalina once again to talk about RNA research.
Awesome. So we focused a lot on DNA. Let's talk about RNA. So we are the only platform where you are able to sequence direct to native RNA molecules. With obviously RNA sequencing, you're getting quantitative and qualitative data in one experiment. You can span the entire length of RNA fragment, achieving like full splice information, differential gene expression, fusion gene detection, obviously RNA-based modification on your native RNA, and many more. Possibilities are limitless. Let's look at some examples of some research that has been done. If you can go to the next slide. Amazing, Steph, thank you. So transcriptome profiling is important in translational cancer research, for instance, and is increasingly finding its way into precision oncology. So while bulk RNA sequencing is widely available, high cost and long data return time are limiting factors for clinical applications. So this this is a study by Andres Mock and Kihan Erkut's team at the German Research Center in Heidelberg, Germany, and it's done with MinION device, the sequencing. So the customers had a few challenges. High cost and long targeted alpha therapy are limiting factors for clinical use of transcriptome profiling of tumors. Studying gene fusions based on short-read transcriptome data is challenging because split reads spanning the fusion breakpoints can carry very limited information about the corresponding genomic coordinates. And also there was a lack of automated bioinformatic pipeline for precision oncology purposes. So here, the shallow nanopore RNA-seq data is compared with short RNA-seq data for 10 samples representing 4 cancer types. Coupled with what they call SHANTI, shallow nanopore sequencing for transcriptomics. It's a newly developed data processing pipeline. They got a turnaround time of just 5 days. There are many advantages Nanopore provided. So long reads, which enabled precise mapping of breakpoints and defining fusion transcript from low coverage, where short reads would struggle to precisely map those breakpoints. They also code a rapid TAT, what I said, of just 5 days. Transcriptomic measurements obtained by the shallow nanopore and Illumina RNA-seq were highly concordant, as you can see in the figure on the right. So they also got transcriptome data where they could infer signaling pathway activities and kinase expression and visualization of those gene fusions. Overall, in what the scientists, in their own words, shallow nanopore RNA-seq, it enabled rapid, cost-effective, and biological meaningful transcriptome profiling of tumors, and warrants further exploration in precision cancer medicine studies. So hopefully, Aaron, me, and Stephanie, we can inspire you to take on the challenge of exploring, rare diseases, cancer medicine, hopefully more with nanopore sequencing. If we can go into the next slide. So we have recently released a full single-cell sequencing protocol that generates full-length cDNA and sequences full-length cDNA from 10x genomic libraries. So you're skipping the fragmentation step required with short-read approaches. So the figure on the left gives you kind of an overview of the library prep of sequencing full-length reads from 10x Genomics. The first 3 steps happens inside the 10x Genomics system, which generates your full-length cDNA. Then there's a step to remove the TSS artifacts from the 10x prep. Then you just prep your libraries, attach our sequencing adapters, and the library is ready to run. So we typically see about over actually 160 million total reads and over 90 million cell and UMI assigned reads with the PromethION flow cell. Awesome. So this is a study that was done collaboratively by Federico Gaetti and Dan Landau from New York Genome Center with our Nanopore New York applications team with PromethION Grid devices. So the whole team had a few challenges. So there can be dysregulated RNA splicing in human hematopoietic malignancies. How this leads to disrupted hematopoietic differentiation, that remains unknown. The inability to distinguish mutant and wild type cells and the cell type complexity of the hematopoietic system, it makes it difficult to see how somatic variant is impacting the transcriptome. Plus, single-cell RNA-seq by short-read sequencing is limited in its ability to map full-length RNA isoforms and uncover those splicing aberrations. So their aim was to simultaneously profile gene expression, cell surface protein markers, somatic mutation genotyping, and RNA splicing within the same single cell. So Federico Gaetti, one of the investigators, one of the main investigators, he developed genotyping of transcriptomes for single-cell transcriptome profiling using Nanopore reads, what he calls GOAT. So GOT with long reads, a single-cell transcriptome profiling and proteogenomics allowed for the simultaneous single-cell profiling of gene expression, the cell surface protein markers, somatic mutation, and RNA splicing that I listed before. With Nanopore long reads, they were able to, it enabled efficient single-cell transcriptome profiling. It also enabled high-resolution splice junction mapping and improved the understanding of those aberrant splicing in the hematological disorders driven by the somatic mutation. They also got a 4-fold increase in the number of junctions per cell detected over short-read data that they had done previously. And they also got greater coverage uniformity across the entire transcript compared to the 3' biased coverage that they had seen in short-read sequencing data. So in brief, the paper presents an expanded multi-omics single-cell toolkit to define the cell type specific impact on somatic mutations on RNA splicing, from like earliest phases of clonal outgrowths to overt neoplasia directly in human samples. I'll move on to next. Thank you. Awesome. So Aaron, I'll give it up to my colleague Aaron to have you go through AAV and some microbial examples.
Thank you, Monalina. And thanks, Stephanie. I think those are really nice overviews for many of those applications, from methylation to adaptive sampling to single cell. And as you heard from several of those case studies, many of those were applied to large genomes like human genomes. So what I hope to share in some of these examples are critters that tend to have smaller genomes, but are applications where we think Oxford Nanopore adds some really nice value, especially to core labs, by enabling quick turnaround time, but also leveraging long reads that enable you to get a more comprehensive picture of some of these small genomes. So the one I'd like to start out with sharing here is our plasmid verification workflow. And, I have fond memories of grad school, which is not too far removed, of preparing constructs and then, having primers and going off to submit that to my core lab for Sanger sequencing primarily, which is, for many, many, many years been the status quo for verifying regions of your plasmids that you're manipulating for a given experiment. So what we think we have here, which is quite a nice workflow, it really helps to enable, it really helps to overcome certain limitations, especially with Sanger sequencing for plasmid workflows. So what I'd like to showcase here is that we have the ability, using the rapid kit as Stephanie described, that basically makes a single cleavage, and then there's no need for primers. Then you can proceed after this barcoding step into sequencing. And what this really enables you to do is sequence the entire construct. So now we're no longer limited to a region that we need primers for, say an insert region or an upper read length limit with Sanger of about 1 kb. In contrast, you can now start to verify the entire constructs. And I hope to make the case that many customers and core labs are adopting this now as a new standard, because there were important regions, especially in the backbone or maybe deletions or dimers, that would just routinely be missed due to limitations with other standard workflows. And then finally, I'll describe how we also have a nice end-to-end workflow, which makes the analysis and sharing a report, for example, from the perspective of a core lab, how you can share these reports easily with the labs that submit to you. Okay, next slide, please, Stephanie. So in a nutshell, how the wet lab side of the preparation would go about. Plasmid DNA extraction, that can really be any standard method, overnight cultures, isolate the DNA, and then the samples could be submitted to the core lab, for example. So in-house, in practice, you could use either the 12 or the 96 barcoding kits that we currently offer through our rapid barcoding kit options. And as Stephanie described, on the right-hand side, you can see a schematic in a nutshell of how this transposase-based reaction goes about. So if your input is your DNA of the plasmids, you do a simple incubation step. And so this transposome complex will randomly cleave a region of the double-stranded circular molecule. And then that rapid cleavage and attachment of barcodes then allows you to pool each one of those individually barcoded samples. And then you would attach the sequencing adapters, and then you would be able to load that library onto your flow cell. So it's a relatively quick workflow in contrast to others that might have very long, hours-long sample preparation times. This really can be achieved in a short amount of time. The other key point I would just like to make here is that in contrast to other NGS platforms, you don't necessarily need to maximize all 96 barcodes at one time. The flow cells are reusable in the sense that you can wash them with a nuclease, and this removes the primary amount of the nucleic acids from the previous library, and then you could run another set of barcodes. So it offers a lot of flexibility, and really this is what helps to enable quick turnaround times. For example, if you wanted to do this routinely each day, to get turnaround times within 24 hours. Thanks, Steph. Here, let's imagine you've pooled 96 plasmid constructs, you've performed the sequencing run, you've let it go for a few hours, and it seems like you have sufficient amount of coverage for each one of those samples. What you could then do is proceed to the analysis tool. And so this clone validation workflow, here you can see a few different screen grabs if you were using the cloud-based interface through Epitome. But we also offer the same workflow. You can find it through GitHub or Epitome Labs if you'd like to run it on a local device or local server. But as you can see here, if you direct your attention on the left-hand side, you would upload your FASTQ files to this clone validation workflow. The parameters, there's just a couple if you want to provide information about what the host background might be, what the expected read length might be, and then you could let the parameter, with those parameter settings, initiate the run. Essentially, what this will do is it will execute a series of commands to create a de novo assembly for each one of those samples. So for example, it will implement Canoe, Tricycler, and then Medaka. And then finally, we use plug this into an open source annotation tool called Planotate. And so what you can see on the right-hand side would be an example of one of those outputs for the clone validation workflow. So what you can see, for example, with barcode number 1, that's around a 3,000 base pair construct, and each one of those colored regions is an annotated feature of that plasmid. What you also receive is a final consensus sequence in FASTA format, as well as a CSV file that has this information as well that you can share. So that's in a nutshell how the end-to-end workflow works, which is a pretty simple and all-in-one type of analysis tool. And then here, I'd just like to share a few examples. It's become quite a popular method, and I think it's really fascinating to see this kind of paradigm shift between just the status quo of checking just the insert region of a plasmid to now doing routine whole plasmid sequence verification with nanopore sequencing. These are a few examples here of customers who have used it. You can see that they've tagged Plasmidsaurus, which does this routinely as a service with nanopore sequencing. But a lot of core labs are also adopting this in-house on campuses. And so there's a few examples here. The one on the left-hand side, I think, just nicely showcases how repeat regions can be sequenced through. So for example, you see these CTG repeats that nanopore sequencing was nicely able to resolve. In the middle one, you see an example of an AAV plasmid sequencing through those ITR regions and generating a nice construct. The top right, I think, is an interesting example where an individual had sequenced their plasmid and the raw reads indicated that it was shorter than expected. And what this is indicating is that a deletion had occurred that likely would have been missed with a short read based approach. Read lengths are not a problem. These can be constructs of any size. So the bottom left example is an example of a 40 kb back sequence. So again, because it's read length agnostic and using our transposase kit, constructs of multiple sizes can all be pooled and sequenced without an issue. So lots of interesting case studies, and we think it's a nice routine workflow that can be easily implemented. And I'll leave with one more case study here. This is a nice publication, again, just sort of demonstrating synthetic biology constructs with nanopore sequencing, similar types of workflows to what we've described. But to me, I think from their figure one, that's a nice comparison where in that plot in the orange bar, they're showing 100% coverage of each construct. And in contrast, Sanger on average is much shorter of a proportion that's generally sequenced. And in fact, the error rates are lower with nanopore sequencing consensus data on average than Sanger sequencing. And again, this is in part because we generate so much data for each of the molecules that you can create a consensus sequence at very high accuracy for these. Okay, and then the last one here I wanted to also highlight, in addition to plasmid sequencing, we've also seen a lot of interest in leveraging long-read nanopore reads for verifying viral vectors that are used, for example, in gene therapy programs. So a common one with regard to adeno-associated viral vectors, you can see a case study here. And so in this example from this group by Philip Tai and his team at the Gene Therapy Center at UMass, is they assessed a few different viral vectors with nanopore sequencing. So they have a little bit of a graphical overview on the left-hand side, but in a nutshell, in the middle column, what I can hopefully direct your attention to is that the entire construct from 5' ITR to 3' ITR, Nanopore is able to read through the entire region. And classically, short reads have really struggled through those ITR regions due to secondary structure and repeats. But in contrast, Nanopore sequences through those ITRs, and that really helps to enable full AAV characterization. And that can be quite important in these programs because AAVs might not sufficiently package properly, or there might be heterogeneity where some molecules are full length and some molecules are truncated, for example. So there's an example on the right-hand side that you can see with a different construct. This is a self-complementary AAV. And what I hope you can see in the pileup plot in the IGV viewer is that there appears to be a hotspot of a truncation event occurring. So that's been validated with nanopore reads as well as other long reads, which is a true event occurring in that viral vector. So regions with truncation hotspots can be visualized at the single molecule resolution with nanopore sequencing. And this is one final example here on the AAV side, a publication from last year in Nature Communications, kind of reiterating what we saw on the previous slide, where they had used our ligation-based kit, a native barcoding kit, on these molecules in order to verify if there were any truncations in these vectors. So it was quite interesting. They have created several different guide RNA designs. So what you'll see on the top is guide design number 4, for example. But they sequenced several guide designs. And what they were able to determine is that what you'll see in the quote on the bottom left is that the sgRNA cassette placement and the orientation, that was actually a critical factor in vector packaging efficiency. So they were able to see that by nanopore sequencing, which helped them to design more effective vectors that had fewer truncations. So now we're seeing this as more of a routine verification tool for core labs that are working again with gene therapy groups, as well as companies that are doing this for trying to determine in their R&D phase if these are proper full-length vectors. Okay, so we'll go through a few final examples here, more on the surveillance and the metagenomics side. And so, as we're all very keenly aware, pandemics have occurred over the years, but nothing quite so impactful in our lifetimes as COVID-19. And for public health labs that have really stepped up to detect newly emerging variants, they've been using more and more sequencing approaches for these applications. So, for example, to understand the origin and geographical spread, and to track newly emerging variants, as well as drug resistance factors. That information is also quite often leveraged for vaccine development. And what I'd like to point out here is that Nanopore sequencing technology, for the attributes that it has as a platform with long reads and portability, it had been used even prior to COVID-19. For example, during the Ebola outbreak in 2014, Zika in Brazil in 2015. And today we've seen in the databases that across the world, there are over 1.5 million SARS-CoV-2 whole genomes that have been sequenced with Nanopore technology. So it really has been a useful tool in the public health and the academic space in generating whole genomes to track emerging variants. This is, in a nutshell, probably the two most common approaches that have been used, which are based on PCR tiling methods across the viral genome. So what I hope you can see on the top, that's a representative genome, say for SARS-CoV-2, about 30,000 nucleotides. And in the middle panel, you can see the classic Arctic workflow. So this is generating amplicons around 400 base pairs in length, and they tile across in 2 primer pools across the viral genome. And in the bottom one, in the the little green rectangles, that's a representation of our Midnight protocol, which really takes advantage of designing longer amplicons so that you need fewer primer sets. So what you can see in the table is really just comparing and contrasting the methods, where Arctic, you would need, say, 98 sets of primers. With Midnight, you would need 29. The amplicon lengths are longer. And so one of the useful features is that, as these viruses continue to mutate, we've seen groups have to constantly update the primer sets because a mutation might impact the primer binding efficiency. So fewer primers that anneal to the genome with the Midnight protocol, as well as compatibility with our rapid kit, really have helped to make it a popular and cost-effective and rapid method for whole viral genome sequencing. But what I hope to highlight here is there are a number of ways that these protocols can easily be adapted to especially different viral genomes. So what we've highlighted here in this table, for example, is not only for SARS-CoV-2, but viral genomes ranging from Ebola to Zika to recent case studies with monkeypox and HIV and adenovirus. What you can see is that the number of amplicons may vary, but the methods are really the same. And so in large part with our standard library preparation kits and analysis workflows, we really think this is a nice way to plug and play, basically just designing different primer sets. And so here you can see all the references for different primers that have been developed for these different protocols with Nanopore whole viral sequencing. And then a couple final ones before we wrap up. As well, long reads really help with bacterial isolate sequencing. And so this is a strong use case we've seen with the technology. And in a nutshell, some of the main features are, if a bacterial isolate is sequenced with short reads, it can be very challenging oftentimes if there are a lot of repetitive regions to assemble it properly. And this can also be problematic when creating a closed and comprehensive genome and determining which genes are associated with, say, a plasmid and which genes are associated with the bacterial chromosome. And so what you can see here are examples where the long reads with Nanopore sequencing really help to enable more comprehensive and full closed genomes that can help to associate, for example, if a mobile element or an antimicrobial resistance marker is again associated with that plasmid or not. This here is an example really just highlighting that it's a relatively simple and straightforward workflow, really not unlike the plasmid verification workflow, leveraging the rapid barcoding kit. And then wash kits can also be used. So an example on the right is from a recent publication, really demonstrating that these type of workflows are effective for bacterial outbreak reconstruction and can be brought down to a nice and competitive cost for generating whole bacterial genomes. What I'd like to leave you with here is really, I think, touching on what Stephanie mentioned at the top of the call, which is technology improvements. Here we're seeing the R10 pore, and if you recall from what Stephanie described, is the longer reader head helps to resolve homopolymer stretches. And predominantly what we would see classically, say, on the R9 flow cells is you can generate really nice closed genomes, but oftentimes short reads would be implemented as a supplement to resolve long homopolymer stretches. Now, in contrast, what we've seen with these technology updates in these R10 flow cells is that that's no longer a systematic issue in long homopolymer stretches. So what I'd like to point your attention to in this study here, recently published in Nature Methods, shows a snapshot of one of the figures. So what we see here are 3 different bacterial genomes. On the y-axis, you can see the indel rates, and on the x-axis, you can see the amount of genome coverage. And so in the green plots, what you can see is based on the R9 chemistry, you would see nice coverage, but it would sort of flatten out and still have some of the systemic indels occurring, which could then be polished and brought down with Nanopore plus Illumina data. Now, in contrast, in the yellow bars, I hope what you can see there is with Nanopore data alone, this really brings it down to a low indel rate just with Nanopore only data. And what you can see on the quotation from the publication on the left-hand side is the authors indicate that they don't see any significant improvement in assembly quality for the R10 flow cells by the addition of any short read polishing. So this indicates that near perfect microbial reference genomes can be obtained basically from this Nanopore only data at approximately a depth of 40x coverage. And then as a final slide here on my end, just showing on the metagenomic sequencing side of things, this too is an area where Nanopore technology enables really rapid turnaround times. But one of the key challenges for metagenomics often is not the sequencing itself, but rather enriching the sample ahead of time or during the sequencing process so that you can reduce the amount of background noise, say from an environmental sample or from a host sample. So here I'll leave you with a table of a few examples that show a few of these different enrichment strategies that have been developed, but still enable rapid turnaround times.
Sorry, interrupting, Aaron. I don't think the slide progressed, so just want to make sure.
Sure. I can see this on my end. How about you, Steph, or anyone else on the call?
Yeah, I can see it. Slide 36.
That's right.
Yeah.
Okay, thanks for checking, Monalina. And so in a nutshell, with this table here, hope to highlight a few of these recent case studies of rapid metagenomics protocols. And what you can see in the column on the right is the turnaround times of under 7 hours. In the bottom example, I think this is a nice case study of a recent preprint demonstrating that with that adaptive sampling approach that Stephanie nicely described, by uploading a bed file of the human genome and depleting for all of those host reads, was able to enrich for bacterial pathogens and antibiotic resistance in just a few hours. So I'll leave you with that and happy to answer any questions on the material that we've covered.
Thanks, Aaron. And then in summary, as Aaron and Monalina have described, nanopore sequencing has shown to be very versatile. We have a wide range of flow cells with various pore counts to suit your output needs. We can sequence short and long fragments with a simple click of a button in our software, and you can pull your samples in a highly multiplex manner. The flow cells have flexible run times, so you can run the flow cells for as long as you need and wash and reuse them, which becomes very efficient and cost effective, so you're only getting the data that you need. And we're also able to manipulate the pores and motor proteins to sequence either at a slower or faster rate, which allows us to improve accuracy and output without making changes to the devices themselves. And this is a complete spectrum of all the sequencing devices that we offer to support your goals. So our Flongle flow cells are our lowest throughput option, producing around 1 to 2 gigabases of data on average. And these were designed for smaller genomes, small amplicons, or they can even be used to QC your library before loading onto our MinION or PromethION flow cells. And our MinION and GridIONs use the same flow cells, which produce on average around 20 to 30 gigabases each, with the theoretical max output of about 50 gigabases. And on the higher throughput end, we have our PromethION flow cells, which generate an average around 150 to 200 gigabases each. So whatever your throughput needs, there's a device for every application. And I should mention that our platforms are completely scalable. So the library prep protocols for the MinION, GridION, and PromethION devices are more or less the same. So you can take what you learn on the smaller devices and apply them directly to the larger ones. This way, you're not starting from scratch if your throughput increases and you need to scale up. So you don't really need to learn anything new between each device. And just to put what we learned into perspective, within a single day, you could go from preparing a sample to obtaining new and exciting data that offers a novel perspective for your projects. It only took you and Ashley's group at Stanford less than 8 hours to sequence a patient and identify a rare genetic mutation using a P48. And in just about 12 hours, you can go from sample to answer with 96 SARS-CoV-2 samples on a GridION. So before we open it up for questions from you all, we wanted to leave you with a question of our own. What will you discover? And with that, that concludes our webinar series. Thank you.
Thanks, Steph and Monalina and Aaron. That's great so far. I know we have at least one question coming in from Dr. Land. I can allow you to talk, Dr. Land, so should be able to unmute yourself.
That's great. Can you hear me?
Yes.
Thank you for this great presentation. I actually have two questions. So I'm sure I get the terms wrong, but your selective sequencing, so discarding sequences so you can enrich for certain sequences.
Yep.
The answer is probably no. But when we do RNA-seq, the vast majority, the sequencing costs pretty much double with Illumina sequencing because of the ribosome RNA depletion step, which we have to do. I'm wondering if that can you deplete? Is your selective sequencing stringent enough that we can deplete ribosome RNA actually on your flow cell, or do we still have to go through that?
I can answer that question.
Okay.
Oh, do you want to go, Steph?
No, go ahead.
So currently the way our adaptive, selective sequencing, we call it adaptive sequencing.
Adaptive, yeah.
So it's currently for double-stranded molecules, so DNA. And we do have some experimental consideration that you need to make. What we have seen internally is if anything's between usually 10 to 20 kb gives you the best fragment length, gives you the best kind of coverage and outcome in terms of the adaptive sequencing. Because cDNA is so small, we recommend anything that's less than 3 kb. The current version of adaptive sampling is not the most suitable approach for that. Okay. But we are testing internally with RNA, but that commercially it's not available yet on our.
Because that would be great. It could save us a lot of costs if we could do that. The second question is washing your flow cell and using it again. Now, I don't do the sequencing myself. The people in my lab do, so maybe they know the answer. But how much washing can you do? Can we use a flow cell for, obviously not forever, but should we start doing washing of flow cells in our lab when we do bacterial genome sequencing?
Yeah, so the flow cell, you can kind of think of a flow cell, as a battery. So there is a finite operational time, which is up to 72 hours. But you can, determine how long you want to stop it. Your sequencing run just based off of your coverage needs, because on our Minnow software, you'll see how many reads and how many gigabases you're getting in real time. So you'll be able to tell just from that, okay, I've got X amount of coverage now. You can stop that flow cell at whatever time point that may be and use our wash kit, which contains the DNAs to flush out your library. And then, theoretically, you should have, let's just say you ran it for 36 hours, you should theoretically have another 36 hours left of run life.
That's great to hear. Thank you.
Yeah, there are some considerations to that. We could follow up in an email because, it is going to depend on, how many active pores you have on the flow cell to start with, how often you're washing and reusing it. But yeah, we can definitely follow up with some more information.
The worst that happens is you just have to keep on sequencing another cell. You're not losing anything.
Right.
Because we usually have plenty of DNA for these experiments.
Yeah. May I ask the reason for you wanting to wash versus probably multiplexing your samples together? I believe you said if this is for bacterial genomes or.
Well, sometimes you don't have enough samples, honestly.
I see.
And other times. We get variable depth of sequencing depth, and we can't always predict what we're going to get. Especially with tuberculosis, the purity of the DNA is really important. So it's possible, and my people would have to answer this, that we don't load as many samples as we could on a flow cell, because we don't want to be in a situation where we don't get a good enough number of reads. And so if we can have the luxury of loading fewer numbers of samples, but then washing the cell and reusing it if we get the reads we want, that potentially could be a cost saving for us.
Mm-hmm.
Makes sense. Aaron, didn't you recently speak about some like end-to-end workflow for TB?
Yes, and so we'd be very interested in comparing our end-to-end workflow with your new, what is it, R4 chips cells? I forget what they're called.
That's right, the R10.4.
Yeah, because I think that we get perfect assemblies. So we get 100% reproducibility from sample to sample for the same TB strain. So it would be really interesting to see, with our what I think are perfect sequences, what the differences are with yours.
Yeah, in the just as a specific preprint that was recently produced by Zaman Iqbal's group, they assessed 151 MTB isolates. And this was on R9, finding really strong concordance with whole genome sequencing, even on the R9 flow cell. So happy to pass that along as well as a resource, as well as the one of the papers we shared.
Well, we know that the R9 by itself is not sufficient for the kind of work that we want to do. But so that's why it would be so interesting to see what happens with our improved workflow and your improved flow cells.
Yeah,
so
sounds great.
Level of quality.
Perfect.
I mean, R9 isn't close, even with a lot of depth. So,
this is DC, where we have countless museums.
Oops. Well,
is that me? I guess that was me. I was not doing anything. That just very strange. Any case, yeah. So sorry about that. Okay, great. So. Poonam, I'm sure we'll reach out to them and get some of their new flow cells. But in the meantime, we'll continue working with our current pipeline because it's working so well.
Awesome.
We're big fans, though. Thank you very much. We're huge fans.
Thank you so much for saying that.
Any other questions?
Yeah, if anybody else has some, they can drop them in the chat, Q&A, raise your hand, whatever it might be. I know I got just a kind of random question, because I always like the willingness that Oxford Nanopore's had to do weird experiments and like talk about different products that sometimes fizzle out, like the old smidge ion that was rumored for a while. And is the Voltrax still a thing for sample prep?
Yeah, so the Voltrax does still exist. Yeah, we actually did come out with a newer version of it that's has PCR capabilities. So for a little while was kind of in a limbo stage because we were upgrading the whole device. But that's now currently available. I mean, there still are some limitations to it because the library prep is transposase based. So it's not necessarily suited for amplicons. It's mainly suited for like genomic DNA. But there are some kits in the pipeline to accept like cDNA, a 16S kit that should be coming out somewhere down the line. So yeah, we made some changes to the device itself. The next step is just kind of making the kits a wider range of kits to suit the Voltrax. Yeah.
Okay, cool. And are there any other rumored things that you're allowed to talk about that may or may not be coming on the horizon?
So while we're on the automation side, I just also wanted to say, obviously, Voltrax, we automated the rapid transposase protocol on it. But if anyone's interested in like ligation kit, higher volume automation, we do have automated our protocols on NGS Hamilton, STAR, and then a few other robots.
Okay, cool.
Since no one else is asking questions, I'm going to just keep on going. So one of the other things we're very interested in is variant calling for unfixed mutations in bacterial populations. So with your improved flow cells, do you have any sense of what level of variance you can detect reliably within a mixed population?
Yeah, it's a really good question, David. I know our applications team is currently assessing that on several different isolates. So that's ongoing on the latest chemistry. But I think as soon as they have some of that benchmarking data, we'll certainly be able to share that.
tuberculosis has a lot of homopolymeric regions that seem to be that we've shown are quite important in drug tolerance and other things, and there's probably many others besides the one we've been studying. So it would be really great if we could look at populations, especially at these homopolymeric regions, and sort of call the different variants.
Yeah, and I know that's a specific application that they're working on with tuberculosis.
Okay.
On the topic of like metagenomics, there is a question in the chat, Aaron. I'm not sure if you're best to answer this. So they're asking, can you expand on the capabilities for metagenomics? What sample types have been run through this technology, and has there been a comparison to short read or PacBio?
Yeah, that's a great question. So there are a lot of metagenomics applications, and again, I think primarily there's two features where the long reads really help to enable in this space. Well, one is assembling metagenome assembled genomes with the long reads, and two is the quick turnaround time. And so many of these applications are, in clinical research contexts. If there's a case of suspected sepsis, if there's a mixed population of viruses, whether that's DNA and RNA. These are all really shotgun metagenomics-based approaches. Just as one recent example, when the monkeypox outbreak was occurring, the National Institute of Health in Portugal received a sample of a suspected case on May 17th, and they just used a shotgun metagenomics approach on that sample with nanopore sequencing to generate the first draft genome, which we saw occur with many of those genomes since it's a relatively large double-stranded DNA virus of around 200 kb. I will try to maybe hone in specifically on your question just because there's so many metagenomics applications, but I will just say in this presentation with the example in Nature Methods, much of their assessment was comparing all those different platforms with the latest R10 chemistry on Zymo mock communities. So there is benchmarking data that's publicly available there that you can download as well, and happy to pass that along.
Awesome. Does anybody else have any additional questions?
Are you working on sort of improved pangenomic software? Because a lot of it gets to be in the analysis, or that's something you're not really focused on. Because there isn't a lot of good software out there. Now that we can get these perfect whole genomes, comparing and illustrating the differences amongst the diversity of strains in tuberculosis, for example, is still a challenge from the software side.
Mm-hmm. Yeah, it's a really good question, David. I don't know if I have a solid answer to that question at the moment, but I'll certainly feed that back, and I'm sure the teams are working on it as they assess with these, because isolates is a big focus currently with the internal teams on the latest chemistry. So any of those software analysis updates, I'll certainly get back to you on.
Thank you.
Any other questions? Okay, well, then I guess thank you everyone who's joined, and thank you Nanopore team for an awesome presentation. We'll hopefully have the recording up in the next couple days, but if anybody has any questions, please reach out to myself, Pat at the Genomics Center, or anybody on the Nanopore team, and we'll be happy to help you out for planning your project or getting you to the best solution. So with that, thank you everybody, and look forward to seeing you at the next seminar.
Thank you, everyone.
Thanks, everyone. Bye.
Bye.
Bye.